Hello, my name is Barbara Roof, and I'm an application scientist here at Maverix Biomics. And I appreciate you all joining us here today so that we can give you an introduction to the platform. What you should be seeing on your screen is our dashboard. This is the main landing page for anyone who has an account on the platform. And what I'd like to do is start by giving you a little overview of the platform so you have a bit of orientation, learn how to navigate through the different sections of the platform. And then what I'll do is actually upload, configure, and launch an analysis so you can see how that goes. Then we'll go through the results, and then I'll demonstrate our data visualizations on the platform. So I'll start here on the dashboard. On the left-hand side, you can see there are different projects. As you click between these different projects and select them, the content on the right-hand side will change. All of the data that's associated with any project, and as well as the processing, is all organized on a per project basis. And this makes it easier to sort through your data and analyses on the platform as you continue to use the platform for additional projects and data sets. So this is true for both what you're seeing on the dashboard as well as what you would find as you move through these main sections of the platform using these tabs at the top. So as an example, if I was to click over here on the Data tab, you would actually see this content here that's associated with that project. And when you're not on the dashboard, you can move between projects by using this menu here at the upper right. So you can select between projects here, and you'll notice that the data content will change. This Welcome project doesn't have any files associated with it, whereas this mRNA-seq project here actually has files that were uploaded by the user, as well as those that were derived from the analyses that were run while they were using that particular project. So if I come back to the dashboard, you can see, as I said at the top, you have these different tabs here that we can move through, and I'll show you. But I wanted to show you one other quick thing from here. If you go to the settings, you'll see what account you're in, and there's also a pane down here that shows you different accounts that you might be associated with. The average user on the platform might just have one account with their projects and analyses in them, but if you did have more than one, you would be able to switch between them right here. You'll notice that if I select this project here, the content on the right-hand side disappears. That's because within this particular account, I'm actually just a user, whereas for this platform demo account, I'm an admin within this account. So I get some additional tools over here on the right-hand side, and from here I can actually grant admin permissions to any other user within the account. I can revoke those permissions, and I can also remove people from the account. One of the really nice things that makes it easy to add colleagues as well as collaborators to your account to share your analyses and data is to be able to add them right from here. So as an admin within this account, I can just input an email address here for a colleague within my lab, for instance, click invite, and then they would get an email that would allow them to click a link and then register on the platform and then have access to this account for collaboration. Going back to the dashboard, excuse me, as I said, moving between these different projects changes the content, and so I'm going to tell you a little bit about this content on this right-hand side. The activities are notifications from the platform. So those are things like a notification that you've started an analysis, as well as when that analysis is complete, so that if you were to log back into your account and come straight to the dashboard, you would know immediately that you had a complete analysis and could start analyzing those different outputs. The shortcuts section is a section where some of the higher level outputs within the platform are located, and these are just bookmarked links to that data within the account. So here we have a link that goes directly to the UCSC genome browser for human data that's been provisioned for this particular account. And then there's also some additional links here. These are heat map visualizations that are part of an analysis that was run here within the account, as well as a table browser, which is a browsable list of data that you can use to drive the genome browser. And I'll show you examples of both of those as we move forward. So moving to the Data tab, as I said, what we're looking here is at all of the files in our project file library that are associated with this particular project that I've selected. So if I was to select a different project, that content would change. Moving back, this is a sortable table, so you can actually sort this by create date, by data type, And this choice here is nice. After an analysis is completed, you might have interest in downloading some of these files to your computer for additional analysis off the platform. You could just click on any row within that table, click download, and that'll download that file directly to your computer for additional analysis off the platform. If you click here, this is the upload files. This is where you would upload files to your account, and this is also project specific, so you would make sure that you had selected the project you wanted, and then you would just click add files and select some files from your computer that you needed to associate with that project. Once your files are queued over here, you could go back and make sure that you've added all the ones you want. If you didn't want to upload a file, you could just cancel from here. And once you've queued all the files that you want to upload, you would just click Start Upload. These will upload directly to the secure cloud account that's behind your particular platform account. And then what that does is allow that data to be associated with your project here, as well as securely stored within your cloud account. There's a 4 GB limit to any particular file. So any individual file that was over 4 GB, you couldn't actually upload through this web interface. But we also offer additional alternative solutions for file uploads, including providing you instructions for third-party FTP clients, as well as instructions for command line upload. So then that allows you to do batch transfers of your data directly into your cloud account, and then that's linked to your account here on the platform, and you can begin using those for your analysis on the platform. So once you've uploaded files, you're ready to move to the stage where you actually begin with the analysis. Before I get to that, what I'll do is show you really briefly these final 2 sections on the platform. The Explore tab, this section within the platform, is where you would see any of the analyses that you had run on the platform. In this case, there's one that's already been run within this project, and it has a status of complete. I'll show you in just a moment when we launch an analysis that you'll be able to see the most recent analysis that we've launched, and it'll have a status of running. And you can actually begin looking at that data as it's coming off from that analysis while it's running. So in real time, you can go, you can check on it and make sure, see how far it's progressed. And any of the outputs that are currently available, you can start looking at and potentially downloading from the platform. This final section here is the Results tab, and this is where some of the final outputs from your analysis will be published. So in this case, it's differential expression, so you can see some of the Cummerbund plots that are outputs from this. And this is a nice place to be able to put some notes that are very specific to this piece of data. So you can see down here at the bottom, somebody's left a note, and once that person has left a note, it relates directly to this piece of data. But in addition, any collaborators or colleagues within the account can leave a comment, and it's a nice way to be able to collaborate within the platform. So now what I'd like to do is actually run through the configuration and launch of an analysis so that you can see how simple that is. From the Explore tab, you would click the Start New Analysis button, and then you would pick the type of analysis that you want to run. So we're going to do, for this demonstration, use the mRNA-seq for differential expression in eukaryotes analysis kit. So I would select that, and from here you can see some information about that analysis as well as the configuration panel for actually starting that. The overview of the analysis includes a description, some additional details of the analysis including references for this particular pipeline, as well as a list of all the bioinformatics tools that are used for this analysis, and we include all their version numbers. So each of the analysis kits on our platform is versioned itself, and we keep track of all of the versions of those tools so that if there are any updates and we incorporate those into our analysis, we increment the version of that analysis so that if you ever did want to go back and repeat that exactly, we would have all of the information about which one you used for your analysis, as well as all the versions of the tools that were used. Finally, there's some benchmark information. So this is the information about the benchmark data set that we used for this analysis. And finally, there's an overview of the main steps within this analysis, which includes information on what type of analysis is being done within that step, as well as some of the outputs that you might expect. So now I'd like to configure and launch an example analysis. So all you would do is begin by giving this an analysis name. So I'm just going to call mine mRNA-seq analysis 1. This label that I give it is what will be associated with that analysis as it moves through the platform. And in addition, this would be the label that you would see in the analysis execution history. I would click Add Sample, and I would begin adding my first sample. So I'm going to call this first one Control, and then I would click Add FASTQ Files. Here you have a choice of selecting paired end or single end runs depending upon the data that you're going to be uploading. If you do have single end runs, you can pick all of the replicates that are associated with this particular control sample all at once from the next pane that you'll be seeing here. For paired end runs, you have to actually select these 2 at a time so that they stay associated with one another throughout the analysis. So for this example, I'm going to pick the paired end reads for my first replicate, and I would just click Add FASTQ Files again and add the second 2. So now I have the paired end reads for 2 different replicates for my control sample, and the final thing that I would need to do is just select whether these came from a stranded or unstranded library. In this instance, it's an unstranded library, so I'm going to leave those unchecked and click Submit. So now I've added my first sample, and now I can add my first condition sample. I'm going to add paired end reads again. And just as with the control sample, I'll go through and for each replicate, I will add the paired end reads as paired selections from this pane. And now I have those 2 selected for this particular sample, and I can just click Submit. Now, once you've added your samples, if you see any problems with these, for instance, you've added the wrong replicate for a sample, or maybe you want to change the name, you can just click Edit. This particular data set that I'm using is a HOX-A1 knockdown, so I'm going to update this name to actually call it HOX-A1 knockdown. And as I said, if you had added an incorrect replicate, you could click this X here, remove that replicate, and add the FASTQ files for the replicate that you actually did mean to add, and then click Update. So now I have 2 samples loaded. The final steps are just 5 different options down here at the bottom. For control sample, you would just click Control. These are the names that you gave them, so it should be pretty straightforward for you to pick the control here. At this point, I will say that for the differential expression analysis that we do in this analysis, we use 2 different bioinformatics tools. So the processing goes through QA, read mapping, and then when it gets to the point where it's going to do differential expression, it splits off and it takes your data through differential expression analysis using CUFDIF, and then also differential expression analysis using DE-seq. For DE-seq, you don't need to pick a control necessarily because the way the analysis runs on the platform is doing all versus all pairwise comparisons between all of the samples in your data set. For CUFDIF, there is a requirement at this stage to pick a control, and then that control is used to compare to all other samples in your analysis. The next thing you need to select is which de novo splice aligner you want to use. So for this mapper selection, we offer the option of picking Top Hat or Star. Top Hat is widely used, highly cited, sort of the gold standard for this type of analysis. Star is a really good tool as well, and in fact, running Star allows you to cut down the overall analysis time because these reads will map much faster. So you could cut your analysis in half by using Star. But we offer this option so that as a user you have a choice for this pre-configured analysis to pick between either one. Platform is the sequencing platform that your sequencing runs come from. So if they're Illumina or Ion Torrent, you can pick that here. And then you pick the reference genome that you want to align your reads to and have the analysis done with. For this case, these are human samples, so that's what I'm going to pick here. But if you did have mouse or rat, for instance, this gives you the option of picking between 2 different reference genome assemblies. So for mouse, you could pick between MM9 and MM10. MM10 is the most recent, but for instance, if you had run an analysis in the past where you had mapped reads to MM9 and you wanted to do a side-by-side comparison with this latest analysis, you might actually choose MM9 at this stage. So you do have those options. And then once you've done that, there's only one final thing you need to pick. For your sequencing run, if a spike in control was used, you would just select that here. So that's it. You would just give this a look over, make sure that you had added all your samples correctly, make sure all your replicates are aligned correctly in terms of the numbers that you picked, and then make sure that these final options here are selected correctly, and then you would click Launch. So this is what I was talking about earlier. As soon as you launch, your analysis becomes part of the analysis execution history. This is the label that you assigned. And you can see that this has a status of running. You can select that immediately and start to see some of the details as the analysis runs on the platform. So currently we're in the analysis configuration step, and this is where it's taking all the FASTQ zip files and it's unzipping those. So once those are unpacked, it takes all of the inputs that you selected when you were doing the configuration and gets the analysis started based on those. So you can see over here on the left-hand side that the analysis configuration step is complete, and we've actually moved on to quality assessment. As you open up any of these steps, you'll get a description, and this was sort of what you were seeing when you were looking at the analysis overview. It's a description of the step, what's happening within that particular step in the analysis, as well as some of the outputs that you might expect. So since this one is running, I'm going to show you as an example one that's already completed. So here's analysis that's completed, and now we can actually take a look at some of the other information that's being published to the platform as the analysis runs. If you click Details for any of these steps, you'll actually see the command line, which includes the list of the different bioinformatics tools that were used for that step, as well as some of the parameters that were passed at the command line. Clicking Output, this allows you to see some of the outputs from that step. So for the quality assessment step, what we do is we begin by doing using FastQC to do a quality assessment of the raw reads. And then based on that, we do some adapter trimming as well as quality filtering. And then we repeat the FastQC so that you have the ability to look at a before and after QA for each of your samples. So this is the before trimming for this particular replicate. And you can see that starting off right off the bat, these actually have a pretty good quality scores across the reads. This is that same sample after the adapter trimming and quality filtering, and you can see there's a slight improvement. If you had fairly poor quality scores, you would see a major improvement by the time you looked at your after filtering and trimming. And by doing this, what we end up with is an optimized set of reads that move forward into the read mapping step. So if I open up the read alignment step, similar to the quality assessment, if I click Details, I'll be able to see what the command line was. So this one was selected top hat for the mapping, and you can see some of those parameters that were passed at the command line. In addition, you get notification from the platform on what's being published. So for this, the BAM files that are part of the read alignment outputs are provisioned into the genome browser that's integrated into your account, and those genome browser tracks can now be viewed right from within the platform. The outputs for read alignment, as I said, the BAM files are actually being deployed to the genome browser in your account, but you have the option to download those if you want, and you can do that right from this screen. In addition, any of these outputs can also be downloaded just by going to the Data tab, making sure you've selected the correct project, and then you can download any of those files from there. If somebody wanted to do a batch download of all of that data, that's an instance where we would set up direct access for you so that you would be able to access the S3 bucket in the Amazon cloud that's behind your account and download things in bulk. You have some mapping statistics for each of your samples. And then one of the final things is looking at these pie charts, and this gives you an overview so you can quickly compare visually between your samples, and you can look at any of these individually and see what's being shown here. And what you're seeing is where your reads were mapping to the annotations within the reference genome. So the majority of them are mapping to exons, but you can sort of get an idea of how many of those reads are mapping to other annotated regions, including introns, intergenic regions, as well as UTR. So just by looking at this, even as the analysis is still in process and hasn't even gotten to a differential expression step, for instance, you would immediately be able to see that all of your samples look pretty much the same. You don't have one that's an outlier, and it just gives you an overall view of what your data looks like as it moves through the analysis. I'm going to jump here to the differential expression step. These are some of the final outputs from this pre-configured analysis on our platform. What you get from this, as I said, we do this differential expression with both CUFDIF as well as DE-seq. So when it goes through the CUFDIF, you'll get all the CUFDIF outputs that you can download in one file. You also get some statistics about the differential expression between these two samples. This is the one where we just had a control and the HOXA1 knockdown. And then you can click on these and see some of the Cummerbund outputs that come as outputs from this particular step within this analysis. So this is the one that I had previously showed you in the results. All of these are published here as well, but once they're in the results section, that's where you could actually add notes and comments and use those for collaboration between other members in your lab or between collaborators at different institutes. So you get these scatter plot, density plot. You can also look at a heat map of this data, and I'm going to show you a heat map in just a moment. In addition, the DE-seq analysis, you get the DE-seq output files, and that's a tarball archive that you can download and open up and have all of the outputs from the DE-seq analysis for this. And then there's the heat map. So this is one of the most exciting ways to start analyzing your data on the platform, looking at the results and paring down the data set from your outputs to actually start looking at what's of most interest to you. So what you see here is a heat map on the left-hand side. For this one, as I said, we just have one column in this heat map because we had one sample versus the control. As you mouse over any of these features within this heat map, clicking on them loads that genomic locus within the genome browser on the right-hand side. So this is integrated together so that the data set that's being used to display this heat map is actually what's driving the integrated genome browser on the right-hand side. As you mouse over any of these rows within this heat map, you'll see a tooltip will pop up that has the sample name, and in this case what it'll show you is what sample you have versus what the reference sample is, or control, depending upon how you have this configured. You'll be able to see the log2 fold change. You can look at the abundance values, and you can also see the p-value for this sample. On the right-hand side, you have access to the UCSC genome browser. This is the same as the public browser, but now this is available integrated within your account. And the great advantage to that is, of course, to be able to view the outputs from your analysis right in the context of the public data sets. So in this case, you can see the read alignments for this, as well as the read coverage. And you can configure this just as you would on the public site by adding additional public data sets and tracks. And you can configure the density of any of the tracks as well as all of the other different configurations that you have available to you for the UCSC genome browser. So in this case, you could add some additional genome browser tracks. As I said, you can change the density so that the configuration in terms of viewing those reads, these tracks, excuse me. And then you can just hit resubmit and then add anything that you would be able to see any of those additional data sets here in context with your private data that you had just run within the tool. Another way that you can view this is the same data set that's underlying this heat map can be viewed in a table browser. So this table view allows you to sort this interactively and view any of the data based on any kind of sorting you want to do. So you could sort this by gene symbol, you can sort it by the abundance values, by the p-value, as well as the log2 fold change between these. And just as with the heat map, clicking on any of these rows within the table will load that within the integrated genome browser here on this side. And one final thing is that for either the heat map or the table view, if you click this downward arrow right next to the settings button, you can download that particular data set directly to a TSV file. So you have a nice tab delimited file with all of this data in it, and you can open that up in Excel and do additional analysis off the platform. So this is one of the great advantages of this type of view. For both the heat map as well as the table view, you have the ability to change a lot of things in the settings panel here. So you can sort your heat map or your table by any of these values. The default is hierarchical clustering, but you can sort by chromosome, p-value, abundance, or log2 fold change. In addition, you can filter that data set. So you might end up with quite a lot of genes in your heat map when you look at your initial default heat map, and what it's done for the default is filter by p-value to look for significance. So all of the genes that were in that initial view have a p-value less than or equal to 0.05. You can change this value. If you had a smaller data set and you wanted to actually increase that, you could remove that, change the value, and you can also add additional filters. So if the data set is too large, you can add additional filters to filter that further. Maybe you want to see all the significant genes within that data set, but you also only want to look at ones that have an absolute value for log 2-fold change greater than 2. And by clicking apply, you can re-render that data set, and it would re-render both in the table view, and you could switch back to heat map. And just hovering over that, you would see that now I've gone from thousands of genes in my data set to 529 genes. This might be a much more reasonable set of data for you to look through off the platform, and this is where you would jump over and actually be able to download just that filtered data set so that you could pursue those genes. So in order to show you some of the more settings that are available here, I'm going to switch over to a different heat map which has quite a bit more data in it and a lot more samples. And this will give you an idea of the sort of customization that you can do for the heat map. So if I open up settings, I wanted to show you one thing really quick. These little icons that are both within the settings pane as well as within the configuration panel when you're setting up your analysis will launch you into our knowledge base and provide some tips and guidelines for how to use the settings. In this case, you could jump over and take a look at how to filter your heat map, and the same is true for the configuration. It'll give you some tips on how you would go about using, what sort of values are expected during the configuration when you're trying to set that up for your analysis. So coming back to the settings pane here, this is where I was mentioning the difference between CuffDiff and DE-seq on our platform. For DE-seq, you have an all versus all pairwise comparison, and for right now, what it's selected for the control is the one that was selected at the time of configuration. But this is where you could switch this to any other sample. If you click apply, you can see over here the heat map re-renders, and now this is the control that's being used for comparison to all the other samples. So now all the samples are being compared to just this control. So if you had more than one control in your data set, you could switch between them and re-render that heat map or table view and be looking at that set of data. Here's another thing that you can do. As I said, you can sort, you can filter, and the one other thing that I think is really pretty slick that we just added as a new feature here is the ability to use gene lists. So for this data set, I had created a few gene lists, but you can also upload new gene lists. So if I had a gene list that I had made that's just a text file, and I could show you what that would look like. So it's just a very simple text file. It just has a list of the symbols for the genes that are in my favorites gene list. In this particular case, it's a subtype signature list of genes. So for the analysis that was done here, they were looking at the signature genes that were associated with the two different subtypes they were looking at in this data set. So this is the list of genes. It's showing me here that all of these are being recognized within the data set. If they weren't, they would show up in red, but you would still be able to add that list because you might want to use it for something else later. So I'm going to call those subtype genes. I can add that. Now I have 3 lists that are available to me. As I select those, you'll see they're each assigned a different color, and if I click apply and then I go back and look at my heat map, what you'll see is that any of these genes that shows up will have a little tick mark next to it, so they'll be marked. That row within the heat map will be marked, and I can actually see in the tooltip here which list that gene is from. And so as you go through, you can see other instances. Here's another, and this is a nice way to be able to look through and easily identify the genes that you're interested in from potentially a previous analysis and be able to see where they lie within your data set here. Another advantage is that once you've uploaded these different gene lists and have them in use, you can use them to sort. So if you were to sort by those, what that does is it moves those all to the very top so that you can see them all together. You can see this gene is actually in 2 of those lists. And if you had a really large data set, maybe you had a huge number of genes in these gene lists that you wanted to review, maybe you had a microarray analysis you've done previously and you had a giant set of genes in a list that you wanted to use, you could actually filter the entire data set by just those sets of genes. And maybe we even want to remove these and just see all of those genes. And now once I've filtered, that's the limit of what I'm looking at now in my heat map, is just that set of genes. And so this is a nice feature to be able to customize that heat map view to see exactly what I want to see and avoid being overwhelmed by all of the novel data that might be there. One final thing, and this is the most customization that you can do here. If you click Custom in the sample pane here, what you can do is actually remove samples. You can add samples. And for each of the samples that so maybe I just want to look at this subset of samples. What I can do is select the reference or control sample that I want to use for each of those individually, so now they don't have to all use the same one. And I can even add samples if I wanted to look at one of these samples, and here I'm comparing it to this control or reference sample, and maybe I also want to see a row where I compare it to something else. And you can also change the column label names, and this is useful if you have added a sample more than once. You have the ability to change the column label so now that these can be distinguished from one another within the map. And once I've done that, if I click Apply, not only do I reduce the set of genes, columns that I'm looking at within my heat map, but I have changed the label so that it's easier for me to identify what the data set is I'm looking at. And of course, as you hover over these, you can actually see which samples are being compared to which reference sample. But this really gives you the ability to customize the heat map and gives you that flexibility both in terms of sorting, filtering, and being able to quickly hone in on the set of genes that you want to look at by using the gene list feature.
I'd like to thank Barbara for today's demonstration of the Maverix Analytic Platform, and to all of you for attending. We hope to see you at another Maverix webinar in the future.